Well, hello. I'd like to go ahead and walk through some material about a project I've been doing some research on, and this is titled Bacterial Chemotaxis Control. And I'm going to walk through some highlights of that. There's an article that's been submitted for publication that goes into more detail, but also talk about how it illustrates an architectural framework and also talk about the creation model. And so that's what I'm showing here is I show an architectural model, one of the ways that I've been looking at chemotaxis. This is a top level presentation picture of the creation model. And this is some of the things that I started to pull out in terms of an architectural framework. And so as I'm going through this presentation, this highlight presentation, hopefully you'll get a chance to see some of what I would like to have you begin to think about. So this paper highlights how biological design and bacterial chemotaxis control shows architectural design features in many areas of biological life that reflect an architectural framework in the creation model. So there's a creation model that I will get to here. And so we're seeing things that are coming up with that. And I'm going to start to be referring to the architectural framework and some of the tenets that come out of that. The study highlights the bacterial chemotaxis control process. It contrasts it with two other types of biological systems, ethanol metabolism and brain function, using a tool called the Systems Modeling Language, SysML, to leverage well-defined and proven engineering tools for architecting, analyzing, and refining complex systems. It first introduces SysML. So this is in the paper. I'm not going to go through this in as much detail in this talk. And the advantages model-based system engineering has for capturing system engineering architecture and allowing for ongoing assessment of how biological systems operate and the clarification of how much model detail is required to reveal a system element function. So it's a top-down process, and you go to the level of detail in the modeling to get to the point where you have a good understanding of the architecture for the things that you're trying to explore. Details of the chemotaxis control process are captured in SysML syntax instead of biological syntax. This approach intentionally enables the analysis of the biological chemotaxis control system from an engineered system point of view. After the functional model is analyzed, the resulting insights and observations can be explored. A biological system architectural framework is proposed with information from chemotaxis control, ethanol metabolism, and brain function. So these are the three things that I've explored, and so that's the reason why I'm focusing on those, and I'll have a chart that will give a context of that in a minute. Both architectural models and a biological system architectural framework can act as a scaffolding to help uncover system function, the relationships of system components and processes, and bioinformatic phenotype and genotype correlation. So there's a fair amount of things that can start to come out, and what ends up being relatively simple, straightforward diagrams. Well, this is a process that I am proposing. It's a derivative of something called the object-oriented system engineering method that is published in various sources in COSI, a system engineering journal and organization has talked about it in various publications. And so what I did is I modified it with this reverse engineering. And so basically what it tries to do, it puts things into a manner in which the object-oriented system engineering method can be used. So there's a couple steps in the beginning that has to take place. First of all, getting the information that is done from biological research, from academic articles and textbooks, cataloging the descriptive information that is given there, and then convert it to prescriptive information, or another term would be put it into requirements language, as if you were going to architect or engineer this type of system. Then you can start to create a high-level functional architecture, and this is where you can start to go into more detail using system architecture tools like SysML, and finally do an evaluation of the architecture. So that's just a reminder what the process is. For this particular example, what we're going to be focusing on is, first of all, what this is a flexible approach, but specifically what we're going to be doing is looking at the chemotaxis control in bacteria, and we'll be looking at a specific type of bacteria, E. coli. And so that's where that context is for thinking about chemotaxis. And there is another paper as well as a talk that I talk about the creation model. And so I'm not going to go over this in much detail, but some observations that you can make, and this is something that's not totally novel for what is being explored here, but it hasn't been put into this level of detail for engineering tools, is there's two triplets. First, there's the triplet talking about the Earth expanse, and then the space expanse. And there's things that are required. First of all, when you need to have the basic things of the Earth squared away before you can get into having life, biological life, and finally having man. So this is the summary of the creation model in SysML. The figure shows a top-level diagram that captures the whole creation week in one diagram. In general, after creating the basic infrastructure of space, time, light, and matter, there are two thrusts seen. First, there is the creation of the physical world. Second, there is the creation of the biological world. There are layers in creation that depend upon one another. By way of analogy, to give a mental image of this, one might imply the open systems interconnect 7-layer model for transmission control protocol. TCP/IP represents an engineering construct that is frequently used for communication for internet protocoling. The creation model shows the multi-layer construct of an architecture with existing rules for how each feature is used. The SysML model is an object-oriented abstraction of the array of details that allow an individual to conceive of the whole without thinking about the underlying parts. So each one of these is, you can think of as a black box. There's a lot more details for each one of these. So details about the research threads can be developed depending on the research questions the individual wants to explore. And so ultimately, what I wanted to explore is to think more about chemotaxis, but also start to think about an architectural framework. And so there's a larger context that is going to be used. So I introduced the method of how, looking from an engineering point of view, how it can be looking at chemotaxis control. I introduced briefly the creation model. Now let's give a context about these 3 biological systems that will be explored. Looking at this chart, thinking in terms of the human organ systems, you can see that there's a lot of missing things here, but these are 3 biological systems that I've explored. So at least I'm going to see where I can go with the information that's related to this. And so first of all, looking at the nervous system, I'll just have a couple charts talking about neuroscience, and for the engineering side, it's a lot of times it's called neuromorphic computing, and I'll briefly talk about that. And there's 2 that are related to digestion. First of all, there's metabolism, and I'm talking about a specific type of metabolism, looking at ethanol metabolism. And the other thing is there's actually bacteria in our gut that actually is part of the digestive process. And so it's a nice synergism type of process where we can actually have good bacteria that's helping the digestive process. So those are the 3 things that I'll talk about, and I'll just have a handful of charts talking about each, and then I'll do a look at trying to think of an architectural framework to think about them as an integral whole. So this is what's called a block definition diagram, and it shows the various parts that is composed of a bacteria is composed of. And the block here in yellow is the one that I'm going to be focusing most on here. So bacterial chemotaxis control has already been discussed. I have another paper in publication that has some details about that. So for what we're going to talk about here, only a high-level review will be done, highlighting the features that could be nominated for biological system architectural framework. Chemotaxis utilizes several capabilities in the bacteria to perform the chemotaxis control process. 3 subfunctions are utilized, and I'll just mention the names, and then the next chart will have a little bit more information on this. Something that's called ligand bonding. Ligand bonding is, you can be thinking of like smelling or tasting, and so there's the physical substance that you're tasting or smelling. That's what we have with ligand bonding. There's also signaling, and the type of signaling is called phosphorylation. And finally, there's adaptation, and the type of adaptation is called methylation. And so additionally, molecular components are available when needed rather than dedicated or hardwired. So what that means is a biological system. It's not like your cell phone, where if a part goes bad, that you have to go get a new cell phone. It actually has the ability to refresh circuits, if you will, from a biological sense. The design shows evidence of being optimized for the specific function is intended to do. There's no evidence of utilizing an over-designed system for this applications. Since bacteria are simple organisms, any biological systems can draw from limited resources. As a result, it is advantageous if there are dual use applications of organism resources, and that's exactly what happens. The architectural model for this system highlights a control and action response use case scenario. I'm not going to, the next slide will show a high level but fairly comprehensive example of the use case diagram. And so the process adapts to a particular chemical concentration level depending on how long a particular input continues. So this is a figure, it's called a use case diagram. And even though it's relatively top level, there's a lot of things that are going on. And I mentioned there was these 3 types of activities, these 3 types of things that are taking place, and I'm trying to illustrate them all at once in this chart. So the chemotaxis control utilizes 3 subfunctions with the chemotaxis architecture. First, there's a chemical sensing via the chemoreceptors, and what they're doing is the term I used before, that's via ligand bonding. So there's a certain type of chemicals that the process, the sensors are tuned to actually sense for, and when they sense, then they can actually pass that information on. Second, there's signal sharing via phosphorylation. And so this is the way that signaling is passed on, that acts as a current, if you will, for those that might be familiar with how things work in electronics, if one considers this function with that type of analogy. And third, there's adaptation to attractant concentration. And the term for that is methylation. So it either turns the gain up, turns the gain down, if you will, when that takes place. Chemotaxis is a bacteria's movement towards or away from chemicals. Chemotaxis control is the mechanism by which chemotaxis takes place. So this part of this presentation talks about E. coli chemotaxis, and the differentiator between this type of chemotaxis and other types of bacterial chemotaxis isn't really seen. I have that developed in some other diagrams that I'm not going to be showing about here. But there is an architecture, and the type of bacteria it actually has what's called multiple flagella. And on the cover, I showed an example of what that would look like, just to give you a context. So E. coli swims in a random walk manner due to counterclockwise filament rotation in each of the flagella, causing the bacterial to do runs. And when it goes in a clockwise rotation, it causes the bacterial to tumble. So when it runs, it goes more or less in a straight direction. Otherwise, it's going in a chaotic fashion where it could be just wandering around its area, sensing concentration. And when there's a concentration, it's going to kick in the control system to move it in the direction where there's a higher concentration of the attractants that it wants to get near and take advantage of. The counterclockwise rotation causes the flagella to combine during a run, so all the propeller, they all propel in the same direction. So the way that I'm showing it in this cover, they're not all combined, so this would be giving a chaotic type of motion. So that's a little bit of a highlight of what you have. So you have a couple of modes of operation that takes place. And some of the characteristics of the chemotaxis control are the following. There's 3 tightly coupled sub-processes, and I already mentioned what those are just a few minutes ago. It's an optimized design for this specific application. It detects for specific chemicals, and there's a handful that it senses, and then the chemotaxis sub-processes instances within a bacteria coordinate their efforts. And so you have a lot of these things that are available, but not necessarily hardwire. A chemoreceptor where the ligand bonding takes place is not necessarily bound to some specific things that are doing phosphorylation or methylation. These are all going on in parallel with things that are made available in an interesting way. So that's a little bit of a highlight of some of the things that I wanted to be mentioning for chemotaxis. And so you could be thinking of a bacteria, it's able to taste or smell attractants, and it moves towards those when they're available. And as that concentration increases, that's going to propel it in that direction. Over a period of time, it's going to get, due to adaptation, it's become less sensitive to that, and it's going to be used to being into that higher concentration. And so these are all things that are in this very simple type of design that are taking place. The second thing, I'm just going to have 2 slides on, we'll talk about metabolism. And so the main point that I want to be making about this, and I'll talk in a little bit more detail about ethanol metabolism, is the fact that it's a 2-step process. And so what I'm trying to show here, there's actually 2 enzymes that break things down from one composition to another, and that happens twice. And so there's 3 products. There's the input product, that's ethanol. There's an intermediate product, which is actually something that you want to not have much because it's actually toxic. And finally, it breaks it down to acetic acid. There are other things that go further along, but this is what I wanted to be focusing on. So it's kind of like having an airline analogy where you have a hub and we have this middle composition that is broken down further by another metabolism engine. And so these are all similar to what I was mentioning for chemotaxis. These aren't dedicated, but each one of these engines has an input and an output that are made available in space in the digestive tract that can actually be part of the digestive process. So that's the first of 2 slides that I wanted to mention. This is also what's called a block definition diagram, trying to show you some of the things that are taking place. And the second one is a use case diagram. And so this is just another way of trying to talk about this process of metabolism and how these things have to be related to one another. And so the user, if you will, the human being that's trying to metabolize ethanol, these are the things that has to take place for that all to occur. And so there thus has to be a linkage of each one of these 3 elements here. So first of all, you have the ADH, the link to ADH to ADLH, and finally the ADLH does its thing. And so these are the two areas that were laid out briefly in the last slide when I talked about the different enzymes. And so now we're seeing the enzymes aligning together to do this two-step metabolism process. So that's a synopsis of 2 of 3. And the next one I'm going to talk about briefly, and it'll take a few more charts, is I'll talk about neuroscience, the human brain function. And I have another paper that and presentation that talks about this in detail. So I'm just going to try and allude to things that give some insight into the organization of this. So there's something called the full compute stack. And so there's 7 elements to that each one of these is a building block to the other one that you can start to figure out function, but there's an organized way that things are laid out. And so with this 7-layer process, it's something like I would argue is similar to what we see in the creation model, that there's steps that day one and the things that take place there have to take place before we can go through the next levels of creation. And so that's actually where things get started. And so this is instead an operational type of scenario versus a creation scenario where things in the human brain, the central nervous system, the sensors and other things are all working together. So I'm not going to talk about the details of these 7 layers, but basically we start from very basic of talking about elements and molecules, getting into harnessing physical phenomena, putting those into functional groupings, having 2 layers of architecture, and then we get into these higher level things when we talk about algorithms, and finally how we would be using those algorithms in applications. So I'll show this chart, and we'll not talk about it in detail, but this is another way of looking at these 7 layers, and this is using something called an activity diagram, where I'm starting to show the process of how these layers engage with one another. And I'll flip back and forth between this chart and the next chart. In another presentation that I have, what I do is I talk about these 4 different types of processes, but I decided to highlight it here to give you an idea for these top 4 layers of this architecture. You start to get differentiated with different types of functions. And so what I'm trying to show is these 4 different types of functions: motor control, sensing, brain computation, and learning. So these are 4 of the main things that take place within the human brain or any animal type of activities as well. There's a layer that really needs to be above this to capture human capability, and I'm not going to talk about that here. Okay, so I talked about these 3 different systems. Next, what I wanted to introduce was to think about architectural framing. And so a framework is a foundation for development. It can be useful for capability development, engineering development, and operation and maintenance. And so these are the things where a framework can actually be used. The example that I talked about for brain function is an example of operation. Engineering and actually implementing is what we can see in the creation model. And there's plenty other things that happen. And the framework is starting to give some idea of what would be the capability that gets us all started along this whole vein. So what are some of the biological architectural framework tenets? I wanted to just spend a few charts just to be thinking about that. And so this is stuff that I really don't have time to go into this in detail, but what I've captured here is I've tried to show some sample categories that from some of the things that I've looked at, that it starts to be obvious that we can see these kind of relationships start to pop out. And so these are the categories. And so with this chart, I'm now looking at these three different types of biological systems, and that's what's labeled over here, these architectural sources. And so now what I started to do is we'll look at some of the features that we can be seeing, and where does that start to point out things where we can see a relationship between these model features and these architectural tenets. And so the output of this type of a process is to try and come up with these different types of tenets. And that's what I'm going to be taking as a takeaway from this part of the presentation, is these types of architectural tenets. So this is just a different way of showing those. And if you think of God the engineer, he had in mind that these are the types of things that he would want to have available for biological life, going all the way back to creation and seeing how things can operate fairly efficiently with the various amount of engineering and programming that is innate to every type of organism. So I really don't have time to go into the details of each one of these architectural tenets, but you can pause the video and look over those in more detail. I may go ahead and record a longer version of this to go through those details, for especially if I see that there's interest from folks that they'd like to see that. So let me just finish up with just 3 more slides. So we looked at 3 different types of biological systems. There's plenty more that needs to be done. And then we can start to make some observations about how there's some commonalities in the way that things are done. One term in engineering parlance and computer science is something called design patterns. We see that taking place. And I created this chart as a way of something that's fairly more accessible to show a relationship between the creation framework, which I started to suggest some architectural tenets, talk about the creation model, and the real major part of what I was trying to think of in terms of a biological system was bacterial chemotaxis. So we've seen that there's a couple different types of tenets from the architectural framework. We see how things start to play out in response to that are captured in the creation model and then implemented in a specific biological system, as I laid out there. So I tried to give 4 examples. Some of these are more than one of these tenets that I tried to capture in this chart. And so, but there are some that have a little bit of similarities. And if I wanted to make this in more details, I would have separated that out into specific, even finer grain details, but I just wanted to leave that there for now. So just to finish up, what have we done in this talk? We've demonstrated a process, a engineering-based process, what I call reverse engineering, object-oriented system engineering method. We looked at biological systems and tried to give some indication of how they leverage the creation model, which is what I'm showing here at a high level. I started to introduce how creating a biological architectural framework is possible. And so I started to do that with the process of what I've done by looking at 3 biological systems. And as I've tried to mention, there is plenty more biological systems to explore. And so this is trying to just give a path forward for how the methodology, and trying to show an example, mostly by focusing on bacterial chemotaxis, but also introducing a couple other types of biological systems. All right, thank you very much.